Hello, everyone. Welcome to Foresight's Molecular Machines Group. Today, we're so lucky to have Dr. Rafik Addou with us. He's an assistant professor at the Department of Material Science and Engineering at the University of Texas at Dallas. And he's written over 90 peer-reviewed publications. And today, we'll hear about bridging research and manufacturing. So, with that being said, thank you so much, Rafik, for joining.
Thank you, and thank you to everyone who joined this seminar. So this is part of the molecular machine group. So actually, currently I'm not active on the molecular machine research, even if I have the tool, especially the scanning tunneling microscope. But I want to present today how we can use surface science tools to bridge the gap or between the research and manufacturing, or how we can go from lab to fabs. So before starting, I want to give not moving. Yeah, I want to give it like a short kind of academic journey. So I'm originally from northeast of Morocco, where I did my bachelor degree in physics. Then I moved to the south of France at Aix-Marseille University to do my master in. Actually, my first master was in kind of theoretical physics and cosmology, astrophysics. Then I switched to physics and nanoscience. Then I started PhD in the northeast of France at Nancy, at the School of Mines, in collaboration also with IMPA Tune in Switzerland. But recently, currently they moved to Zurich. After my PhD, I moved to University of South Florida, Department of Physics, to work with Professor Matthias Basel on the growth of graphene and the characterization of graphene on different substrates. Then I moved as a research scientist at UT Dallas with Professor Bob Wallace, who retired last year. I stayed there for 5 years, working with the SRC centers. And the hot topic, as I will show today, at that time was like the growth and the characterization of other 2D materials. After 5 years, I moved to Oregon State University in the Department of Chemical Engineering. As you see, like from the name, I was doing a lot of actually catalysis, but still using the same technique, surface science technique. Then from 2021 to 2024, I moved to Ottawa to work for Canadian banknote nanotechnology, like a company for 3 years. And since summer 2024, I moved back to to UT Dallas as assistant professor. So now it's like almost it's 1 year and a half exactly here at UTD, at the Department of Material Science and Engineering. So today I will present like why surfaces and interfaces are interesting, and why we study them like in general, and also what kind of techniques that we use for to study and explore surfaces and interfaces. Then I will present the cluster and or my labs, because I have one lab. I call it like the cluster lab, and the other one is the XPS STM lab. And I will give some examples for exploring low-dimensional materials, including like thin film with different surface science like metrology, and the majority of them they were related to the SRC project that I was like leading here at UTD, which I was working closely with companies like Texas Instrument, Intel, Micron, IBM, Global Foundries. Those are some examples of studies. Then we'll finish with the summary. So the bulk of crystals or and materials mainly provides the mechanical support and and acts as a reservoir of atoms and charge, while surfaces and interfaces are actually where almost all the interaction with the outside world take place. So these surfaces, interfaces determine the physical and the chemical properties of the materials. And at these boundaries, materials exchange charge, mass, and energy with their environment, and control transport barrier, initiate mass reaction, and Filer mechanism. As devices start to shrinking, actually, to the nanoscale, the surface to volume ratio grows so large, it grows so large that interfacial properties, means noble properties, become the primary determinant of performance. So the surface and interfaces, when we shrink materials, becomes the primary determinant of the performance. What does it mean? This means practically, this means that, for example, in transistors, the gate dielectric semiconductor and metal semiconductor interface set the threshold voltage, the mobility, and leakage far more than the deep bulk of the channel. Another example, for example, in catalysis, only surface atom at active sites participate in reactions. The rest of the bulk mainly just works as support. In batteries or electrochemical systems, charge transfers, degradation, the kinetics are all governed at the interface between the electrode and the electrolyte. So even thought that the bulk stores the ions in the case of batteries. This is kind of the progress of surface science through the more than 100 years. So in modern devices, the bulk provide, as I said, the structural stability, but the surfaces and interfaces drive the functionality, means drive the application. So the progress of surface science is fascinating journey that spans more than a century. It begins with the study of solids in the early of the last century, when William Bragg introduced X-ray diffractions in 1913, earning the Nobel Prize just 2 years later. This era, called also the electrification era, laid the foundation for understanding crystal structure and band theory, and this is thanks to the pioneer like Felix Bloch with his theory in 1932. As we move forward, the focus shifts from the bulk to the surface, and with the Ernest Ruska's invention of the transmission electron microscope in 1932, opened new possibilities for imaging at atomic scale. By 1946, John Bardeen introduced surface state theory, which was critical for understanding electronic properties at surfaces. Then one of the breakthrough is 1954, with Kai Siban developed the X-ray photoelectron spectroscopy, which is a technique that revolutionized chemical analysis of surfaces. Then Sims in 1982 for chemical analysis, as well as STM and IFM in the early 18s. These tools marked the transition into the information era, from where surfaces now and interfaces become the central to the technology. And one of the technique that I will present today, which is scanning tunneling microscopy, developed by Binig and Rohr, who invited from Switzerland. With this technique, enables direct visualization of atoms and earning them the Nobel Prize. So this shifts from solids to surfaces and interfaces reflect a profound truth captured by Pauli's famous quote: "If God made solids, surfaces were the work of the devil." So surfaces are really complex, boundaries, and full of surprises, yet they are the key to modern science and technology. For many years, surfaces were considered complex and poorly defined, so the introduction of ultra-high vacuum system fundamentally changed these perceptions. So UHV made it possible to prepare clean, well-controlled surfaces and study them reproducibly, transforming surface science into a rigorous and quantitative discipline. So at the nanoscale, a large fraction of atoms sit at the surfaces and the interfaces. So electronic, chemical, mechanic, and all the properties are dominated by the surface structure and its chemistry. So solving the issue of fabrication and integration issues, which are actually critical to realizing the full potential of devices based in 2D materials. So for example, this schematic illustrate the importance of understanding and controlling interfaces within different component of device. And you can see there are a lot of interfaces in this structure or in this device, between the gate, and the channel, the source and the channel, the gate oxide and the channel, and also between the channel and substrate. So all those interfaces, they drive the functionality of the device. So to study these surfaces and interfaces, we need to look at them at the nanoscale. For example, using this smart chart from olefins, we will find a wide range of techniques for material characterization. Where's my precursor? Yeah, yeah. Now you can see my precursor in a wide range of material characterization technique from the surface to the bulk. And you can see that here is the surface. Here we can see all the microscopy technique, which are shown here, the imaging technique. And as we go, we in more in this direction, it's the sensitivity of each technique. For example, if you take, for example, let's see, FTIR, it's sensitive to, or the detection limit, it's 100 part per million. For example, 0.1 atomic percent, you can find like FTIR and also OJ. If you want to go like more sensitive to the chemical sensitivity to the surface, you need to do, for example, EELS, OJ. And also you need to find the right tool to, if you want to study like up to 3 to 10 nanometer below the surface, you have technique like XPS and OJ. Just the topmost surface layer, you have techniques like atomic force microscope or optical microscope, scanning tunneling microscope, or TOF-SIMS. And as you go deep, you can find like more techniques. Raman is up to 1 micrometer below the surface. RBS also 1 micrometer below the surface. FTIR 1 micrometer below the surface. Then if you go more to the bulk, there are techniques like XRD or SIMS. And some of them, for example, TOF-SIMS, you can also do dynamic SIMS. So you can have like etching at the same time. You are like, but in this, all the techniques with the arrow means that you can do sputtering, and in this case you will also it's damage your initial surface or your initial material. So so this is another factor to select the right technique. So we have like three different kind of pillars: microscopy, spectroscopy, and diffraction. So when we have a study that we want to do or a question that we need to answer, so we need to think of surface science not as a single experiment, but a kind of puzzle. So we need to investigate the material. We need to look at surfaces and interfaces where actually every atom counts. And no single tool actually exists that can give you the full answer or the full picture. So to solve an experiment or puzzle, we need a combination of microscopy, spectroscopy, and diffraction techniques. Microscopy will give you kind of the eyes, whether either STM, IFM, TM, or SCM. We will see the physical landscape, the terraces, the steps, the defect, the atomic arrangement. Spectroscopy will provide you with the identity and the chemical bound, the elemental analysis, or the concentration technique like XPS, OJ, or Raman. Diffraction will reveal the order and the arrangement technique like XRD, lead, or read. So the real breakthrough happens when these techniques complement each other. So you may see feature with microscopy, you identify that feature and its chemistry using spectroscopy technique, and you can confirm the symmetry and using diffraction. In my talk, I will focus mainly on two techniques that I have on my labs, and they are in the same system. The STM, which will provide high resolution images of surfaces, which it's a great technique to study surface structure and electronic properties at atomic level. For the people new to this field, so how it works, you have sharp metallic tip, which is brought extremely close to a conductive or semiconductive surface. And when a voltage is applied, electrons tunnels through the vacuum gap. Then a quantum mechanical phenomenon, tunneling current is measured, which is exponential sensitive to the distance. And as the tip scans, it adjusts its height to keep the current constant and mapping the surface atom. This is how we can use tunneling current to use it as and as fundamental for scanning tuning a microscope, and you can imagine that we can use other properties for to use them for other mode of IFM. Like we can, if we measure, if we care about magnetic properties, we can have like magnetic force microscope MFM, or work function. So we have like Kelvin probe force microscope, or electrostatic. So we have different mode for the atomic force microscope. This is kind of microscopy technique. Now, if in the side of the spectroscopy technique that I have is the XPS, which provide chemical composition information, which include composition like chemical bonding, concentration. It's a useful technique like to analyze surface contamination, identify elements, their concentration and their chemical bonds, how it works. The sample is irradiated with X-rays. So those X-rays photons are absorbed by the core level electrons, which are then ejected from the top 1 to 10 nanometer, because the X-rays, the energy of the X-rays that I have magnesium and aluminum, so I can go down to 10 nanometer below the surface. So then we will measure the kinetic energy of those photoelectrons, and we can relate the kinetic energy to the bonding energy of those electrons, which give us information about the elemental and the chemical analysis. And also we can have valence band maximum, and we measure also the work function and other properties of materials. So I have the first lab, which we call cluster lab. So this is an image of this lab. So it's not clear here what we have, but in this schematic, you can see that we have a different deposition chamber and an analysis chamber. In terms of deposition, we can do atomic layer deposition. For now, we have hafnium oxide, aluminum oxide, zirconium oxide, and titanium oxide. We have the precursors to deposit these materials. Here we have PVD chamber. Here we deposit metal by evaporations, and we have in the same chamber up to 8 metals. We have like one evaporator where we have like four low work function metals like titanium, chromium, scandium, and the other side we have four high work function metals like gold, palladium, platinum. So we do the deposition here. This small chamber here, we call it annealing chamber, but here we do some kind of annealing heating. We can also do some other processing like annealing in not only in USV, but in oxygen environment, in ozone environment, nitrogen environment, hydrogen environment. We can do also UV ozone. Here it is another deposition chamber, but it's by sputtering. So we have up to 4 targets that we can use to sputter materials. So for now we have hafnium, titanium, tungsten, and tantalum. But we have in the storage like more than 25 targets. The last chamber is an MBE chamber, and this is dedicated to deposit materials from the column where we have like we can do silicon, germanium, and carbon. And all these are connected through this tube, this transfer tube, with pressure in ultra high vacuum and exactly low 10 to minus 11 millibar, and everything goes through this transfer tube to go to the analysis chamber where we have an XPS, UPS, OJ, an ISS, and also lead. So we can do elemental analysis of and chemical analysis of our deposition, our surfaces and interfaces. We can sometimes we, for example, even if we want to do deposition of a metal on the top of a high K dielectric, so we can do that here without going like without taking the sample outside. This 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 tool in the beginning had an STM here, as you can see here. This is the wobble stick to take the sample from the XPS chamber. But since they moved this in 2000, I think, 13 to a first floor, so then the STM never worked. Yeah, because of the vibration. Now we have here a lead. Yeah, and I will show you next the other tool that I have that has an STM, which is like a miniature of the cluster tool. This one has only two chambers. We have deposition chamber here with the lead and the quadrupole mass spectrometry, and where we can deposit materials. We do lead, we do annealing, then we transfer to the analysis chamber, which is here. This one is the analysis chamber, where we have an XPS with monochromator and also non-monochromator, aluminum and magnesium for XPS. And we have an VT STM in the analysis chamber next to the XPS. This is the one that I was using actually at Oregon State University, which is kind of similar to this one, but at Oregon State University and at the chemical engineering department, they were doing a lot of catalysis. So this one is near ambient pressure XPS and near ambient pressure STM. So the STM you can run STM in this up to 100 torr, and in XPS here you can go to up to 10 to minus 3 millibar. As you can see, the detector is here, but the cell where we do like the sample is here, and we have here like 3 different differential pumping, because we are going like from 10, like, sorry, I said 10 to minus 4 millibar, no, up to 10 millibar, 10 millibar, but we work, yeah, 10 millibar. So we go from 10 millibar to ultra high vacuum through these 3 different differential pumping.
We
But
can see the cursor, by the way.
what is it? Yeah.
We can't see your cursor.
What about now?
No.
I think there's a way in Zoom to get like a laser pointer thingy.
Oh.
You see your slides, just not what you're pointing at.
Pointer options, yeah, automatic.
Try the little pencil in the bottom left.
Yeah,
Mm,
I did.
I see a cursor now. Yep.
yeah.
Okay.
Cool.
Oh,
But now
Yeah.
it disappeared. It comes and goes.
I can see it from my side, but
Oh, I see it now.
yeah, I need to keep moving it in.
Oh, okay.
Meaning- Yeah, so so this one is a kind of surface science tool dream, where actually you can have side of where you can do deposition of materials, and the other side is the analysis tool. So for example, especially for this one, those are 3 different MBA, but you can have like an MBA with CVD, with an ILD, with even like connected to glove box. This is the loud lock. And here you have low temperature STM here, and here you have an XPS chamber. And this side, this one was designed to have an HR eels here in this side. But this is just kind of, if you want really to investigate or to answer the like a fundamental question, you need really to have this combination and those complementary technique, like microscopy, spectroscopy, and also diffraction. Now I will start to give you some kind of example of what I was doing like in the past and also currently. Actually, what I like to do, or the focus of what I'm doing now, it's really when you see the technology readiness level chart, you can see for each like technology, you always start with the basic technology, which is which is TRL 1. And when this technology is mature, now you are reaching like system operation, and now it's in the market. What I like to do a lot, because my tools allows to do a lot of fundamental research, fundamental understanding. I like for either novel materials or any materials for any technology, to do a lot of fundamental understanding and engineering and understanding on atomic level. And this can go from TRL 1 up to TRL 5, means basic technology research, research to prove feasibility, and also technology development. Doesn't mean that I don't work with. I have some project actually where the technology is mature, but they still need those tools, for example, to understand if they have issues in the reliability or failure issues. So they go back to understand what is happening actually at the interfaces. For this, I was doing in the past a lot of for any technology, you need to have materials, and the materials you need to grow it. So when you are growing like new materials, you need to tune the parameters to get like your best materials in terms of defect and also of scale, and also understand the interfaces. And this is one of the work that I did actually for graphene. And this is the growth of graphene on platinum 111. So we can form this kind of moire pattern because there is a mismatch between the unit cell of graphene and the platinum 111. And you can see here there is grain boundary and different defect. And here is the atomic scale, the structure of the graphene with some point defect. And the lead shows those 6 spots here belong to the graphene, and this kind of arcs because the graphene is rotating with different grains, so it has different unit. Each grain, you see this grain and these grains, they have different unit cells. So and this form different moire that you can also this symmetry in the lead. For MOS2, when we started with MOS2, before even people they started to grow it, we were because molybdenum disulfide exists in minerals. You can find rocks. There are mines in Australia, in Canada. There are a lot. China, they have mines of MOS2 molybdenite. So when you see at atomic scale, you see like kind of perfect surface termination of the sulfur. We can see that, but actually, when you zoom out, you see that there are a lot of defects on these materials. So one of our goal in the past is to kind of inspect the quality of the materials are used in 2D field effect transistors. And we were actually providing that, but using the scanning tuning microscope, atomic force microscope, and also X-ray photoelectron spectroscopy XPS. Then when we found that, because how do we started this? Because we were seeing like different papers reporting different kind of mobilities and the different performance using like the same material. So we started to investigate that, and we found like from sample to another sample, the quality is really different. So we went from we did a lot of investigation. For example, here you can see that in the same MOS2, or even later I will show tungsten selenide or other materials on the same material, on the same surface, you can find different type of defect, and the defect density can go like a very high, up to 8%. Some defects are really very large, like 3 to 5 nanometer, and they are different. We can find metallic defect, donor and acceptors. We can find single sulfur vacancies or chalcogen vacancies if we are talking about tungsten telluride or tungsten selenide, and there are too many structural defects, and this will definitely impact the electronic and the physical properties. And here another example kind of other TMD materials, TMD for transition metal dichalcogenides. This is like geological MOS2, and this is synthetic MOS2 grown by chemical vapor transport, tungsten sulfide, hafnium selenide, moly selenide, rhenium sulfide, moly telluride, and tungsten selenide. And when we want to see how this will impact, you can see when we doing like scanning, tuning, spectroscopy to measure, like our goal is actually to measure the band gap. But from a region to another region, we can see that there is really a lot of variation in terms of like the position of the Fermi level is changing, the size of the band gap, the defect state inside the band gap also. So there is a lot of variation. And this will definitely explain why from paper to another paper, why the performance is different. So here, using STM and STS, we are at the nanoscale. But if we want to go a little bit high in scale, so we did XPS. And those XPS, they were where the spot size is around 500 micrometer. So what we did is, let's correlate nanoscale with the microscale, means, and means how we will do that is actually we put a mask on the MOS2, and on the same hole we can do an IV measurement, electrical measurement. We can do STM, we can do STS, and we can do PS on the same hole. So we can correlate the electrical measurement with the chemical characterization and the morphology and the electronic property. So as you can see, when we have like an area with low density of defect, we can find that we have a Fermi level close to the conduction band. So it's an N-type MOS2 as expected. And we can also see only single peak, means that we have only one chemical state of molybdenum. which is molybdenum bonded to sulfur. And because this one was n-type, so we see that is from the IV curve, it's also n-type behavior of this MOS2 in this hole. So we just moved away by around 1 millimeter. So we pick another hole, and we find that now it's p-type behavior from the IV curve. The the density of defects is really high. There is change in the STS, and also in the XPS. We are detecting second peak. So in the N-type MOS2, we have only one peak at 229.1 EV, but this one we have an additional peak which is shifted by around 0.8 EV. And when we measure the stoichiometry for each region, for the N-type MOS2, we found the ratio sulfur to moly around 1.8, and for the P-type MOS2, sulfur to moly is around 2.3. Means it's sulfur rich, and here it's sulfur poor. So then we changed it to synthetic tungsten selenide, and it was not that better. It's not like that good than MOS2. We also see a lot of defects and also a lot of variation. And we want to see if this, for example, is it just surface because of the exfoliation or because during the growth. So for that, we did kind of high resolution Rutherford backscattering. This will give us actually the stoichiometry of the sample, but not only on the surface. The surface is here at 0, but up to, for example, for this sample, up to 80 nanometer below the surface, and for this sample of hafnium selenide, up to 120 nanometer below the surface. So we can understand some kind of variation near the surface in the stoichiometry, probably due to the exfoliation. But as you can see between these two samples, so C and A is not for amorphous and crystalline. Just we want to, because we bought an MOS2 where from the Australian mines, and another MOS2 from Canadian mines, and we can see that in terms of the concentration of the molybdenum, it's, if you focus on the one in the red one, so it's more sulfur rich than the other one, which is from Canadian mine. So those MOS2, they show variation not only on the surface but also inside the bulk. This hafnium selenide it's not a mineral, but it was grown by chemical vapor transport, and we did the same technique to see the variation. So, and as you can see, the ratio is close to 2, but in a region like around 300 angstrom below the surface, we can see like there is a huge defect. It's very hafnium deficient, and there is a lot of selenium, and also there is a variation from, yeah, not close to, no, if you see the variation here, it's much stronger than the variation around 100 to 200 strong below the surface. So this was another technique like this to complement our what we found from XPS, because the stoichiometry that we get from XPS is only up to 10 nanometer below the surface. If you remember, we had a lot of kind of local variation in our STM samples. As STM image, we see a lot of local variation, but we believe it's due to the presence of impurities. But when we do XPS, we cannot find any impurities. And we know that XPS is sensitive, or the detection limit of XPS is one per thousand, or 0.1 atomic percent. So for that, we need a technique that can detect impurities below this value. So we did ICP-MS technique, which actually we can get the concentration in part per million or part per billion. So we use the company, Balaz Lab. So we send them different samples, and they did, because they work a lot with the semiconductor industry. So they do this a lot for the company. They use silicon for that. This is the why they pick only the element they study for semiconductor industry, and they don't pick the element that the semiconductor company they don't care about. So for that, you see that some elements that, for example, iron, tungsten, silver, cadmium, lead, bismuth, antimony, are really at very high density with higher than 10 to 12 per centimeter squared. And those, if present in silicon, this impurity, if present in silicon, they will induce levels in the band gap, especially the red ones here. This is MOS2. We hope that if we do synthetic MOS2, where we control the purity of our precursors, so we can get better, like more clean samples, which is true that we have less element detected with the synthetic MOS2, but there is still some element. If present in silicon, they will introduce like a defective in the band gap, like tungsten, cadmium, antimony, bismuth, and lead, and others. The one are in gray are below the detection limit of ICP-MS. And as I said, when we receive the data from Bala's lab, we want to see, okay, we have those elements are really in very high concentration. Actually, what we did, we went through. So when you do a survey, which is like kind of low resolution, so you only detect the main peaks here. They are labeled here because they are core level for molybdenum and sulfur, and also some oxygen and carbon as contamination on the surface. Then we scanned each region, but high resolution XPS to see if we can find those elements like from the ICP-MS measurements. And we didn't find nothing. So yeah, so all those element impurities are below the XPS detection limit, but they are present in ICP-MS. But since our STM is very local, so when we measure like a local, so indeed we can see variation in the contrast in STM, but there is no explanation from XPS. So this is just summary for other TMDs that we did, tungsten selenide, tungsten sulfide, tungsten telluride. Yeah. So a lot of growers in the world, they need someone to kind give them inspect their materials and give them like the quality of their materials. For that, they always use like the tools that they are present, especially like in the clean room. You can find like scanning, electron microscope, XRD, XPS2, the IFM. And for that, we worked with different people, growers across the US, and especially Josh Robinson from Penn State University, Chris Henkel from Notre Dame University, and also Rudney Roth when he was at UT Austin before he moved to South Korea. So they were growing samples, and they want to know what are the properties of those materials, what are the quality of the surfaces and the interfaces. So we were doing a lot of STM, XPS, lead, and this is an example actually of the growth of the graphene on nickel 111. And in this study, we found that there is always competition between the growth of graphene and carbide, because our goal here is to find what is the minimum temperature that we can grow graphene on nickel 111. But at certain temperatures, especially around 500 C, you don't grow graphene, but you grow carbide. So we did this study with scanning, tunneling microscope, lead. We have OG data, but this is low energy electron microscopy, LEAM, and it was done in Brookhaven National Lab. So we can take like kind of those are screenshots from a video. So we record videos of how the growth. So you can see like from at 550 C. Actually, we start with the carbide. This is the transition temperature from carbide to graphene. We start actually with carbide because we see this signal from OJ that is representative of carbide. But with time, this carbide is transforming to graphene. And when we do OJ in this, for example, especially at higher than 500 C, we can see we get this signal which is specific and characteristic of graphene. What we found too, that for certain condition we can actually control the growth of bilayer graphene. So we have the first layer here, but if we keep leaking, we were using ethylene. So if we keep leaking ethylene after the formation of the first layer, and especially if the orientation for certain orientation with the nickel 111 surface, we can actually have this allows to the formation of second layer of graphene. And we can see it here for the lead. We have 6 spot for the first layer, and since the second layer is rotated in respect to the first layer, so you can see that we have another 6 spot in the diffraction pattern, showing the symmetry of the second layer of graphene. So some of the work that we did with Josh Robinson in Penn State University. So he was like one of the first that he was growing tungsten selenide using CVD and more CVD. And the substrate here is graphene ground on silicon carbide. And you see this is high resolution STM image of tungsten selenide, showing the symmetry, the right unit cell, the right band gap for monolayer. And also here you can see the diffraction pattern of tungsten selenide, and this is diffraction pattern of graphene. And there is now like it's not oriented. They are they have like the same orientation between the top layer of tungsten selenide and the under layer of graphene. To verify that, we did XPS and we detect like core level of tungsten and selenium with the right stoichiometry. This is also actually was the first time that Chris Henkel trying to grow hafnium selenide. Hafnium selenide, it's much reactive and oxidize very fast when it going in air, because this one was shipped like from Penn State to UTD, and we can see some oxygen, but we don't see really like tungsten oxide or selenium oxide. But this one, at that time, Chris Henkel, before he moves to Notre Dame University, was here at UTD. So it's just the lab next to the XPS and STM. So even if we do the transfer less than 5 minutes, we can still detect hafnium oxide on the top of hafnium selenide. And when we take it also the STM, it's hard to image really the surface because the formation of the oxide on the top, but we managed to get atomic resolution of hafnium selenide with the right band gap of 1.1 eV, which is expected for bulk hafnium selenide because this is bulk material ground by MPE. And we need other characterization technique to get like the valence band edge. This will give us the position of the Fermi level and also the work function measurement. Then we can construct a band diagram of this material. I will not go through all what we did, but also we did for other materials ground by MPE, tungsten telluride, bismuth selenide, and also alloys. For ILD, this was done at Oregon State University in collaboration where they want with the collaboration with the electrical engineering department, where they want to, this is the first time they tried to use this precursor here to grow tungsten sulfide using H2S. So, and they needed help to give them if they are really growing tungsten sulfide and what is the level of tungsten oxide or what are the other like contamination or impurities. So we did XPS to characterize that, and this is the core level for tungsten sulfide, and also this is the core level of oxygen 1s that will give them how much tungsten oxide or tungsten oxygen nitride they have in their film, so they can tune the parameters and find what is the best recipe to get large and pure tungsten sulfide. This is like first time growing tungsten sulfide by ALD using this precursor.
I just want to flag that we have about 5 minutes left.
5 minutes left.
Yeah. So maybe we want
Yeah.
to open it up for questions, or if there is outstanding information that you think you'd like to share, I'll do it
What
up to
they
you.
can do, there are some, yeah, I can wrap up in 1 minute or two.
Okay.
Yeah. So probably I will go directly to, this is some metal contact that we were doing. Like we can do metal contact for devices here and we do the characterization here. And we showed what is the difference if we do the metal contact in situ or we do the metal contact in the clear room. And there is and as you will see in this comparison, ultra high vacuum deposition, high vacuum, you don't get the same interface chemistry. And probably I will probably finish with one recent work that I'm doing, which is really not, I can do with, when you have surface science lab, you can do research at nanoscale level, but also you can go to the space. So for that, currently I'm doing developing some materials for space coating. especially for satellites in low Earth orbit, because there is a lot of atomic oxygen at this altitude, and there is a lot of like degradation of the materials, and also because of the degradation of the materials, there is also drag. And for that, I'm developing some kind of metal oxide to coat satellites to reduce like that will withstand atomic oxygen and also reduce the drag. With this, the satellites can stay longer in low Earth orbit. So in as summary, like surfaces and interfaces of low-dimensional material, this is what I do for living, and this can be applied to different kind of fields in chemistry, physics, electrical engineering, chemical engineering, bioscience, mechanical engineering, nuclear science. So I worked with all those departments. I did work for Intel, HP, Nike, and having those tools, you can control and understand what is happening at surfaces and the interfaces. And also I did some other that I was not able to cover in this presentation about like passivation, catalysis, corrosion, and functionalization. This is the team, my current team at UTD. And thank you also to the sponsors.
Thank you so much. Let's quickly move into questions. If anyone has a question, I think we have time for 2. So you can put it in the chat or raise your little Zoom hand, whatever you prefer. Okay, Tad, you're up.
Hi, thanks for the interesting variety of surface measurement techniques you described. So one of the ones I was particularly interested in is if on the surface we had moving molecular machines, say of a nanometer type scale, and we wanted to check whether they move the way we hope or how much force they exert, which of these techniques that you've described might be particularly helpful for that? And conversely, are there things that we could do to modify the machines that would make them easier to measure their behavior if they're on a surface?
This will need more like atomic force microscope, which I don't have in my lab, but I have collaborated. They have atomic force microscope because you are talking about the force. So not using like a scanning tuning microscope, but we need an atomic force microscope. If you have like single 2 nanometer or 3 nanometer molecular machine, and you wanna like see kind of how fast, for example, we can probably use STM to measure that. We need just to take snaps. Especially now, there is a lot of they are developing really fast STM that fast,
To be helpful,
but
yeah.
not in picosecond or nanosecond. Yeah, fast in terms like if currently my images, I'm running my images at probably 15 minutes per image. For example, the STM that I was using at Oregon State Universities, we were taking the full image for probably 10 to 15 seconds.
Mhm.
Yeah, but to understand the force, really you need an atomic force microscope, not scanning tuning microscope.
Yeah, well, as you've described, having a variety of tools is helpful, and that's why I appreciate that.
Yeah, like the force, the force. Yeah, it's called, I think, the force, or yeah, FMM. I think the force microscope. Yeah.
Yeah, thank you very much. I want to go to go to the next person question.
Awesome. I think Micah will be our last question, if you want to go, Micah.
If anyone else has one, you're probably better than mine, so speak up now. I'm just curious, the engineer in me in particular is very curious. How do you troubleshoot things like the tip of an atomic force microscope? Do you have like another atomic force microscope that you use to measure the tip of one that might be broken? Like when you're troubleshooting a problem, how do you address that when you're working on these things that are supposed to measure very small things?
Can you say it again? Using the tip in the
Just-
atomic.
Yeah, how do you troubleshoot problems with a microscope that is designed to measure very small things? Like the tip of the atomic force microscope itself is so small that you would need another atomic force microscope to like measure it if there's a problem. Like how do you deal with problems when you're building these?
For example, for the tip, if we know is the issue with the tip, we just change the tip.
I see. Okay. And so,
Yeah. But if something like in the electronic, there is other thing that we can do. But if it's the tip itself, yeah, we will need just to change it. Or before we change it, we do a lot of prep to make sure if we can save it, if we can make it sharp, for example. So, for example, we can sometimes we go under the surface by 1 or 3 nanometer and we pull and we see if we can make it sharp. Sometimes we apply high bias for example, if there is any contamination that will drop on the surface, so we clean for example, the tip.
Interesting. Thank you.
Well, I think that will conclude our very short Q&A. But I want to thank you, Dr. Addou, for your amazing presentation
you,
and for
everyone.
joining us. It was such a pleasure to have you.
Thank you.
And for everyone else, we will see you here next month. So hope you have a great rest of your day.